Hi, everybody. My name is Dr. Ashley Moore, and today we're going to be talking about the stages of neurodevelopment, specifically as it relates to our prenatal development. So obviously we start with simply a fertilized egg, a sperm and an ova, and this is called a zygote. And so in order for this to become a human being, different things must happen. Because a zygote will divide into 2 cells, and then those 2 cells will divide into 4 cells, and those 4 cells will divide into 8 cells. But if that just continues, we'll just have a blob of cells. So there's additional steps that need to happen for this to form an actual human being. So the first thing that must happen is the cells must differentiate or specialize into the type of cells that they will become. And we've talked about this previously in our previous video. But in addition, the differentiated cells need to align with similar cells in order for them to become structures, such as organs in our bodies. And also cells, specifically neurons, have to establish functional relationships with other cells. So they need to synapse so that they can communicate with one another. And so basically, this is the process that needs to happen for just a bundle of cells to become a human. And this tends to happen prenatally, so in the womb. So first, the differentiated cells that will form our nervous system become the neural plate. Then these neural cells proliferate. Then they migrate and aggregate. Then the axons grow and form synapses. And then finally, there's death of some of these neurons and rearrangement. So we're going to go through each of these 5 steps and talk about what's happening in each. So the first stage is the development of the neural plate. And the fetus at this point is composed of 3 layers of tissues, the ectoderm, the mesoderm, and the endoderm. And the ectoderm forms a cluster of cells on it called the neural plate. It's often depicted in a way that looks to me like an oyster. It's a flat surface that will become our entire nervous system. And so these cells are differentiating into our nervous system. The neural plate has an interesting way of developing. And so here, what you see is 2 images. The top shows the neural plate as it folds, as the 2 sides begin to fold up. The picture below it shows a cross-section of the neural plate, so you can kind of see it from 2 different angles. But after a few weeks, what happens is the neural plates, the edges of the neural plate kind of start to fold up on each other. And at this point, the lower portion down here is called the neural groove, and the upper portions up here are called the neural crest. So you really just have a groove at this point down the length of the fetus, which will technically be the spinal cord. Once the lips of that groove begin to touch each other, then they start to fuse. And once these have fused, they really create an enclosed section called the neural tube. And this is what will eventually become our spinal cord, as well as the ventricles in our brain. So it's basically the system that's carrying our cerebrospinal fluid is what this enclosed tube is. And there are certain disorders that can happen during this time of development. So one of the main ones is spina bifida. And spina bifida is when one end of this neural tube does not fuse completely, and so there's actually an opening. But in normal development, this would entirely fuse and create an enclosed space that was not accessible by any sort of outside orifice. It was completely enclosed. So once the neural tube is formed, what's known as neural proliferation starts to happen. And proliferation just means basically an expansion in number. And so neurons and neural cells start to increase greatly at this point. We produce many more neurons than we will actually use, in fact, which we'll talk about in a few minutes. This is not completely uniform. So, you've got a tube, and if the cells expanded in a uniform way, we would just be a very tubular looking fetus. And we kind of start out that way, as in the picture here at 25 days. But as you can see, at 40 days, we have many more, bumps in certain areas where you can tell neurons are proliferating more in specific areas. What will become the cerebrum, this pink area, is actually where we have the most proliferation. And so this actually creates some of the characteristic patterns of folding that we see in the nervous system of different animals. Is that proliferation determines how the shape actually takes place. The third stage is neuron migration and aggregation. So in migration, the cells, once they're formed, need to move to the place where they will eventually have their final location. And usually we think about this in two types of migration. There's radial and tangential. So radial migration moves from the neural tube outwards towards the outside of the fetus, whereas tangential migration can be in a circular fashion or along the length of the fetus. And these two processes happen in slightly different ways. So tangential migration occurs by a process that's called somal translocation. You can think of sort of the way that a worm moves or the way that a snail moves in terms of, let's move over here out of frame. So the snail would sort of extend its body and then pull itself to that position and then extend its body and pull itself to that position. That's very similar to what neurons are doing in somal translocation. They have an extension that extends out and the backside of the body is pulled towards that extension. And that continues as it moves throughout the body. On the other hand, radial migration is usually mediated by what is known as radial glial cells. These are a type of glial cell that form a organized structure. You can kind of think of it as like a ladder that allows these cells to follow these glial cells to their location, usually in a sort of a straight pattern. Aggregation, instead of migration, refers to cells joining together and sort of clumping or adhering to form larger structures. So maybe the heart or the cortex of the brain or specific regions of the brain. And this is usually done through cell adhesion molecules or CAMs. These are actually located on the surface of the cell. They're proteins there, and they allow cells to recognize similar cells and adhere to one another. And so this is, in addition to migration and aggregation, this together forms sort of the third stage of neural development. The fourth stage of neural development is axon growth and synapse formation. So once all of our cells in our nervous system are in place, in the place that they will continue to reside, then they need to start to grow their axons. Remember those long connections that connect to other neurons. And of course, they need to know which other neurons to connect to. So this is quite an important process. Growth cones are what is sort of observed on the tips of these growing axons. And this is thought to be a chemical guide to where they are supposed to be, their intended targets. Astrocytes are another mechanism by which axons figure out where they're supposed to be and where to form new synapses. So although this process is not entirely understood, growth cones and astrocytes do appear to help the axons figure out where they're going to end up and exactly which cells to synapse with. And then in the fifth stage, which is really the final stage of prenatal development, is neuron death and synapse rearrangement. So we talked about how during proliferation, you make a lot of cells that you don't necessarily need. We make a lot more cells, about 50% more cells than we will actually need. So once the cells are identified that are not needed, a lot of those die in apoptosis, which we've talked about before. In addition to just the cells dying, the synapses for the cells that remain alive may decrease in number. So sometimes this is called synaptic pruning. They think of the same way you would prune a rose bush. You might prune the dendrites of a neuron in order to decrease or change the synapses. And these processes are, while they may seem a little bit counterintuitive, they're thought to make the brain more efficient. So it's getting rid of things that are redundant or not needed within the brain to make the processing faster and more efficient. So I've also included a sixth stage, which is myelination. This doesn't necessarily occur so much prenatally, it's mostly after birth, but it is an important stage of brain development. So just as a little bit of background, between birth and adulthood, the brain pretty much quadruples in size, and a lot of this happens in the first 3 years. Now, a lot of people think this is because new neurons are being produced, but that's not really the case. There are three things that are really responsible for this increase in size in brain. The first two are related. They're synaptogenesis and the branching of dendrites. So the neurons that are currently in our brain, the axons may branch further, form more dendrites, and synapse with other neurons. And this creates some additional brain size, brain volume. But the main mechanism behind the increased size is myelination. And we've talked a little bit about this earlier, but myelin is that fatty sheath that wraps around the neurons, and it increases the speed of the electrical current through the axons of the neuron. And so if you are familiar with electrical wiring, you'll know that this is also a long wire with sort of a insulated piece of material around the wire. A lot of people think of axons and myelin as the wire as the axon and the myelin as the insulation, which both have the same function, which is increasing the speed of electrical conduction down the wire or the axon. And so this process is happening all through our adult lives, and our prefrontal cortex, responsible for planning and decision making, isn't fully developed and myelinated until our early 20s. So this really goes on quite a bit postnatally after birth, and is really kind of the sixth stage of our neural development. So hopefully that gives you a little bit more insight into the different stages of neural development and what's happening in each stage. That's all I have for today, and hopefully I'll see you in our next video. Bye.